Good day, everyone. We are the group 3, and our topic is all about specific host defense mechanisms and introduction to immunology. So, for the introduction, immunology, when we say immunology, it is the scientific study of the immune system and how the body defends itself from the diseases and the infections. So, nag-focus ni siya kung paunsa ang atong body mag-recognize of harmful substances and also mag-develop po ni siya of protection sa atong body. So, scientists who study this field are called immunologists. Next is for the overview. The immune system is considered the third line of defense in our body. It provides a specific defense mechanism, meaning mag-react lang siya sa certain pathogens or foreign objects nga nag-enter sa atong body. So it acts when the first, the skin, the mucus, and the second, the inflammation or phagocytes lines of defense fail. So next is for the historical background. So I know we are all familiar with this. So immunology started with early scientists who made discoveries about how the body fights diseases and how vaccines work. So first here is Edward Jenner. So Edward Jenner, we all know that he developed the first vaccine in 1796 using cowpox para makaprotect sa mga tao from smallpox. So this was the beginning of immunization. So next is si Louis Pasteur. Louis Pasteur, siya ang nag-prove nga ang microorganisms makakause of diseases and develop vaccines for anthrax, cholera, and rabies, leading to modern immunology. So for Bering and Kitasato, in 1890, they discovered that the body produces substances or antibodies in the blood that can neutralize toxins. This explained how immunity works. So for the last is Michnikov. Michnikov, he discovered phagocytes, white blood cells that eat and destroy harmful microbes, showing the body's cellular defense mechanisms. Next is for the key to understanding immunology. So for understanding immunology, it focuses on two main terms, the antigens and the antibodies. So for antigens, mao ni siya ang molecules nga nag-trigger sa immune system to respond, while ang antibodies are proteins made by the immune system to fight those antigens. For the differentiation between self and non-self, the immune system identifies the body's own cells as self to avoid attacking them. When it detects something unfamiliar like bacteria, viruses, or toxins, it labels them as non-self and starts a defense response.
For this part of our report, let's begin with the major arms of the immune system. The immune system has two major arms that work together to protect our body from harmful microorganisms. These are the humoral immunity, also called antibody-mediated immunity, and the cell-mediated immunity, or CMI for short. Both of these arms defend the body from specific pathogens, meaning they target particular invaders instead of providing general protection. However, they differ in how they achieve protection. In humoral immunity, protection is achieved through antibodies that circulate in our body fluids, such as blood and lymph. In cell-mediated immunity, the protection is carried out by immune cells themselves, which directly attack and destroy infected or abnormal cells. So while both aim for the same goal, defense against infection, they use different strategies to do it. Now let's talk more about humoral immunity, also known as antibody-mediated immunity or AMI. This type of immunity always involves the production of antibodies in response to the presence of antigens. Remember, antigens are substances like bacteria, viruses, or toxins that trigger an immune response. Once antibodies are produced, they stay and circulate in the blood plasma, lymph, and even body secretions such as saliva and mucus. Their job is to recognize, bind to, and neutralize the same specific pathogen that originally stimulated their production. In humoral immunity, the antibodies are the main players. The immune cells play only a supporting role, if any. To summarize, humoral immunity provides protection by using circulating antibodies that target invaders outside of the body cells. For example, bacteria or toxins present in the blood or tissue fluids. Moving on, let's now discuss cell-mediated immunity, often abbreviated as CMI. Unlike humoral immunity, cell-mediated immunity does not primarily rely on antibodies. Instead, it involves the direct action of immune cells, mainly T lymphocytes, as well as macrophages and other specialized cells. In this process, these immune cells identify and destroy cells that are infected, abnormal, or foreign to the body. That's why antibodies play little to no role in CMI. Cell-mediated immunity is especially important for defense against intracellular pathogens such as viruses and certain fungi, because these microbes hide inside our body cells where antibodies cannot reach them. It is also vital in destroying tumor cells and in rejecting transplanted tissues since the immune cells can recognize these as foreign. So to remember it easily, humoral immunity fights threats outside the cell, while cell-mediated immunity fights threats inside the cell. Next, let's have an overview of immunity in general. The term immunity refers to the body's resistance to certain infectious disease. When a person is immune, it means their body can fight off or prevent infection from occurring. Interestingly, humans are naturally immune to some diseases that affect animals. This is because our cells lack the specific receptors that those pathogens need to enter and infect us, or because of other complex natural resistance mechanisms built into our biology. There are two main types of immunity based on how it's obtained. Innate immunity, also called natural immunity, which we are born with; acquired immunity, which we develop during our lifetime, either by being exposed to an infection or through other means. In short, innate equals inborn protection, while acquired equals learned protection or developed over time. Now, focusing on acquired immunity, this is the type of immunity that develops after birth as a result of the body's exposure to antigens or through the transfer of antibodies from another source. There are two main types of acquired immunity: active acquired immunity, where a person's own body produces antibodies after being exposed to an antigen; passive acquired immunity, where the person receives ready-made antibodies from another person or animal. The key difference lies in who produces the antibodies. In active immunity, your body does the work itself, which makes the protection long-lasting or sometimes permanent. In passive immunity, you only receive the antibodies that others need, so the protection is immediate but temporary. Let's take a closer look at active acquired immunity. This type of immunity happens when your own body is stimulated to produce antibodies. It has two forms: natural active acquired immunity, which occurs when a person becomes infected by a live pathogen. For example, when someone recovers from measles or chickenpox, Their immune system remembers that infection and develops long-lasting or even permanent protection against it. Artificial active acquired immunity, which happens through vaccination. Vaccines contain weakened or inactivated pathogens that stimulate the immune system to produce antibodies without causing the actual disease. Examples include vaccines for polio and rabies. In both forms, the end result is the same. Your body develops protective antibodies that can recognize and fight off future infections by the same pathogen. Finally, let's discuss passive acquired immunity. This type of immunity occurs when a person receives preformed antibodies made by another individual or animal. Because the body doesn't have to produce these antibodies itself, the protection is immediate, but it lasts only for a short period of time. There are two forms of passive immunity: natural passive acquired immunity, which happens naturally. For example, when a fetus receives maternal antibodies through the placenta before birth, or from colostrum and breast milk after birth. These maternal antibodies help protect the baby until its own immune system matures. Natural passive acquired immunity, which happens naturally. For example, when a fetus receives maternal antibodies through the placenta before birth or from colostrum and breast milk after birth. These maternal antibodies help protect the baby until its own immune system matures. Artificial passive acquired immunity, which involves injection of anti-serum or immune globulin, such as rabies immune globulin, or anti-venom used after snake bites. In summary, passive immunity provides fast but temporary protection, while active immunity provides slow but long-lasting defense.
And now, let's proceed to the cells of the immune system. Immune system involves complex interactions among many different types of cells and cellular secretions. These cells work together to protect the body from pathogens and maintain the overall health. The example of this is when you get a cut or nagkaroon kag sugat sa other areas of your body, ang immune cells is mag-rush siya punta dun sa site para ma-prevent ang infection. And immune system is meron siyang two main categories. The first one is lymphocyte and the other supporting cells. Lymphocytes, ito yung type of white blood cells na naa siya important role sa ato ang adaptive immunity. Unlike innate immune cells, lymphocytes can recognize and remember specific pathogens, allowing for a more targeted and effective response upon subsequent encounters. And ang example ani is, for example, naa kay chickenpox virus. Then, ang trabaho ng B cells sa atong lymphocytes is, i-recognize niya yung chickenpox virus, then mag-create siya antibodies, providing immunity for years. Then, ang T-cells naman, if natay cancers, ang T-cells, gina-attack niyo yung cancer cells doon sa tumor. So, ang natural killer cells naman, ginadestroy niya ang virus na infected cells without a prior exposures. And ang atuang lymphocytes is meron siyang three main types. Ang B cells, T cells, and natural killer cells. And each one of it is naa siya unique functions. So, let's proceed sa B cells. B cells are responsible for humoral immunity, which involves the production of antibodies. When a B cell encounters an antigen, it differentiates into a plasma cell, a specialized cells that produces and secretes large quantities of antibodies. These antibodies circulate in the bloodstream, neutralizing pathogen and making them for destruction by other immune cells. So, kaning B cells daw, mature into plasma cells. Then, yung plasma cells mag-produce ng antibodies. Then, yung antibodies, in-neutralize niya yung pathogens and mark them for destruction. Ang example, ani, is kanang nagpa-vaccine ka. Tapos, after vaccinations, ang B cells mag-produce siya antibodies against the weakened pathogens, then providing protection against future infections. Next is T cells. T cells are central to cellular immunity, which involves the direct killing of infected cells and the coordination of other immune responses. There are several types of T cells, each with distinct function. Helper T cells assist other immune cells by releasing cytokines, while cytotoxic T cells directly kill infected cells. Regulatory cells, T cells, help to suppress immune responses, preventing autoimmunity. So, yung helper T cells daw, ina-assist niya yung other immune cells. Then, yung cytotoxic T cells, ginakill niya yung infected cells. While yung regulatory T cells naman daw, sinosuppress niya yung immune response to prevent autoimmunity. Ang example ani is kanang ang T cells, ang helper T cells coordinating an immune response against the flu virus by releasing cytokines. Cytotoxic T cells directly killing cells infected with the herpes simplex virus. Regulatory T cells preventing an autoimmune reactions by suppressing T cells activity against the body's own tissues. Kaniya ang types of lymphocytes. And this one is the features of the functions of our T cells. Next is the plasma cell and antibody secretion. Plasma cells are highly specialized cells that are dedicated to producing and secreting antibodies. They are differentiated B cells that have undergone clonal expansion and somatic hypermutations to produce high-affinity antibodies. These antibodies bind to specific antigen, forming immune complexes that can be cleared by phagocytes or active the complement system. Plasma cells daw, gina-differentiate niya ang B cells. Secrete large quantities of antibody. Then, antibodies mag-bind siya sa specific antigens. Ang example ani is kanang yung plasma cells rapidly producing and releasing IgE antibodies during an allergic reaction to pollen. Next one is the macrophages and dendritic cells. Antigen presentation. Macrophages and dendritic cells are antigen-presenting cells that play a critical role in initiating adaptive immune responses. These cells captures and processes antigens, then present them to T cells. These interactions activates T cells, leading to development of cellular and humoral immunity. So, yung antigen presenting cells, ginakapture and process niya yung antigen. Then, naga-initiate po siya adaptive immune response, present antigens to T cells. Then, ang example ani is kanang yung dendritic cells, ang engulfing bacterium in the skin and presenting its antigen to T cells in a nearby lymph node, initiating an adaptive immune response. Next is natural killer cells. Non-specific cytotoxic activity. Natural killer cells are a type of lymphocytes that provides rapid, non-specific cytotoxic activity. Unlike T cells, natural killer cells do not require prior sensitization to kill infected or cancerous cells. Natural killer cells recognize target cells through a variety of receptors and release cytotoxic granules to induce cell death. So, kaning natural killer cells daw, nag-provide siya rapid, non-specific cytotoxic activity and then targeted, infected, or cancerous cell. And nag-release siya cytotoxic granules to induce cell death. And ang example ani is kanang natural killer cells detecting and killing a cancerous cells that has lost its MHC class 1 molecules preventing tumor growth. And the next one is the other supporting cells. Mast cells, basophils, eosinophils, monocytes. In addition to lymphocytes, there are several types of immune cells that play important roles in inflammation and immunity. These include mast cells, basophils, eosinophils, and monocytes. Mast cells and basophils are involved in allergic reactions and inflammation, while eosinophils target parasites and contribute to allergic inflammation. Monocytes differentiate into macrophages and dendritic cells, further supporting immune response. So, other supporting cells daw, yung mast cells involved in allergic reactions and inflammation. Yung basophils naman, nag-release siya histamine and other medications. Then, yung eosinophils naman is gina-target niya yung parasites and nag-contribute siya sa allergic inflammation. Then, yung monocyte, gina-differentiate into macrophages and dendritic cells. So, ang example ani is kanang yung mast cells daw, nag-release siya histamine during a bee sting causing localized swelling and itching. Then yung basophils naman, contributing to airway inflammation in asthma by releasing leukotrienes. Ang eosinophils naman is targeting and killing parasitic worm in the intestine. Then yung monocyte, differentiating into macrophages in lungs to clear inhaled dust and debris.
So let's now move on to immune system structures and sites of immune responses. So we all know that the immune system is like, diba, the body's army, the body's defense system. And our defense system actually consists of organs, cells, and tissues working together to defend our body from pathogens like germs or bacteria and viruses. Now, the lymphoid organs are categorized into two. But before that, ano ulit yung lymphoid organs? So, lymphoid organs are like kanang parts or parang mura siya warehouse kung saan ginagawa or tinitrain and ina-activate yung mga immune cells aka lymphocytes. So first we have primary or central lymphoid organs. This is where the lymphocytes develop and mature. And secondary or peripheral lymphoid organs kung saan dito nagmi-meet yung immune cells aka lymphocytes, nagmi-meet yung lymphocytes and germs, and then they fight each other. Two categories of organs. We have the primary and secondary. So the bone marrow, its function or role is it makes blood cells and helps B cells mature. And thymus naman, it is where T cells grow and learn to not attack the body. So next one, we have the secondary organs. We have the lymph nodes, spleen, tonsil, and malt. So what does malt mean? MALT. It is mucosa-associated lymphoid tissue. Please remember, So lymph nodes, they filter tissue fluid. Basta lymph, it is tissue fluid. They filter tissue fluid and trap germs. Next one, spleen. Spleen filters the blood and detects germs in the bloodstream. So later on, we will learn more about spleen. So next one, tonsils. Tonsils. So basically, it protects the mouth and nose area. Next one, or lastly, the mouth. And the mouth protects the inner surfaces like the gut, produces immunoglobulin A antibodies. Now, where do immune responses occur? So first, we have spleen, tapos lymph nodes, and mouth. So, kanina, di ba, we tackled about the spleen. It filters blood, but aside from that, spleen also attacks germs that are found in the blood. Now we have here lymph nodes. Again, they attack germs from tissues or wounds. And mouth, they attack germs that enter through the nose, mouth, or gut, either inhaled or ingested. Now in these examples, we will learn how the body responds to germs in different places depending on where the germ enters. So first example, we have tonsils and adenoids. So they defend against inhaled or intranasal antigens. In simple terms, they defend against germs we breathe in. Again, basta tonsils and adenoids, they defend against the germs we breathe in. Second example, we have the Pierce patches sa intestines. So they defend against ingested antigens held by M cells, aka microfold cells, that transport antigens to these tissues. So Pierce patches, in simple words, they defend against germs we swallow in. Again, we swallow in. It may be food or water. Okay, circulation of lymphocytes. So we have to keep in mind that lymphocytes, aka immune cells, they are like para silang yung mga police na nagpapatrol. So they ensure surveillance throughout the body, and every antigen is likely to meet its specific lymphocyte. So they start off as naive. So they are not activated yet. Number 2, they travel through the blood. Tapos sa secondary lymphoid organs. Ano nga yung lymphoid organs, diba? Lymph nodes, spleen, tonsil, and mucosa-associated lymphoid tissue. And after that, they travel to the lymph, tapos back to blood. Third, they enter lymph nodes through special vessels called HEV, or high endothelial venules. So number 4, once they meet a germ, they activate and become fighter cells, or aka memory cells, then move to the infection site. So what is the purpose of this circulation of lymphocytes? So its purpose is simply to make sure that the immune system can detect germs anywhere in the body.
Humoral immunity. So what is humoral immunity, guys? So, ang humoral immunity is an adaptive immune system that is mediated by antibodies. So, what is antibodies na pod? Antibodies is ang molecules composed of carbohydrates and protein. In short, it's a glycoprotein, good. And it is produced by B cells in response to antigen. In many cases, these antibodies are capable of recognizing, binding, and inactivating or destroying specific pathogens. Next one, what is antigen? Before I explain what is antigen, as you can see on the photo here, here. As you can see, kaning color yellow, mo na ang pathogen. And the pathogen, ang surface sa kanang pathogen is dira nakagapuyo si pathogen. So, okay. What is pathogen? Antigen. Antigen, so wait lang. In short, ang pathogen is a part of antigen. Ang antigen is a part of pathogen. Just ko learn. Antigen is our foreign organic substance that stimulated antibody production. It generates antibody production, making it an antibody generating substance. substance capable of stimulating antibody production are antigenic or immunogenic. So in short, ang antigens is ano siya, antibody generating substance or mo ni siya ang stimulator to our immune system. So what is the importance of antigen? Antigens are important because they trigger the immune system to create a response, protecting the body from foreign threats like viruses and bacteria. And we can use antigens in medicine practice like vaccines development triggers immune system para ma-activate ang antibodies and create a memory. So syempre pag ma-recognize na or kanang ma-recognize na sa atong mga antibodies sa atong first encounter, so the second encounter nato sa kaning specific pathogens is dili na siya maka-affect sa ato. Why? Because na recognize na nagkaroon na ng memories ang antibodies sa katong specific pathogens. Okay. So, we're here. These are the type of antigens, or antigens can be proteins that more than 10,000 DA or Daltons. Mangutana mo, unsa na ang Daltons? Ang Daltons is mo ni siya ang molecular weight. Yun ang currency for molecular weight. And antigens can be a polysaccharides, a combination of biochemical molecules such as glycoproteins, lipoproteins, nucleoproteins, and also a large molecules of RNA or DNA. And they can be a cellular components of microorganism or macroorganism such as helminths. And a notes that antigens are foreign proteins are best antigen. So next one is the antigenic determinants. The antigenic determinants are also known as epitopes, are specific sites on antigens where antibodies bind. And also the kaning epitopes are mo ni siya ang individual molecules that stimulate for production of antibodies and referred to as antigenic determinants or epitopes. Same raman. A bacterial cell that can describe as mosaic of antigen determinants and haptens. Wait lang, before haptens, certainly that all invading microbes fall into these categories. Some small molecules called haptens kaning na dire may act as antigens only if they are coupled with a large carrier molecule such as protein. So if isa ra ang haptens, then they cannot trigger our immune response. So unsa na sila? Charing. So antigens pa ba? If mangutana mo, antigens pa ba ang haptens? The answer is incomplete antigen. They can be if mabound man gud sila sa sa specific molecules or protein like immunogen. For example, ang penicillin molecules may act as haptens, causing more people to become allergic or hypersensitive to them. Like gamay man gud ning penicillin, so kailangan nila makabound. The reason nga nung most people is allergic to penicillin, it's because ang kaning penicillin is hapten sila, then if makabound sila sa specific protein sa atong lawas is mag-cause jud na sila og allergic sa ato. Okay, the next one is the antigen processing in the bodies. The processing of T-dependent, wait lang. Okay, dira sa T-dependent and T-independent antigens. Okay, so let's for antibodies to be produced, antigen must be processed within the body. So this involves the macrophage, T cells, B cells working together. Antigens processing is complex series of events. Okay, let's explain the macrophage here. Macrophage engulf and digest and digest antigens. And the B cells is recognize processed antigens and help activate the B cells, while the B cells is developed into plasma cells that produce antibodies. So in short, si B cells yun ang most dire kailangan nato in order for us to produce an antibodies. Okay, so next one here is the T-dependent and T-independent antigens. So what are the difference between these two? So ang T-dependent antigens are required helper T cells, macrophage, B cells for processing. And the T-dependent antigens only required B cells for processing often large polymeric molecules like polysaccharides. So mangutan na mo if independent man ni si T independent is because dili na niya kailangan ang T cells. In short, B cells siya iyang kailangan. Because ang T-independent antigens are unique in their ability to activate B cells directly to their structure and strong signals they generate through extensive BCR cross-linking. So, without the helper T cells. So here, as we can see here in the table, in the figure 16.3. So, first up, dira ta sa taas explain. So, kaning B cells with specific antigen receptor is nagbind siya sa kaning isa ka antigens. Here, kita nyo sa kanang circle, magbind siya. So, pag magbind na siya, ma-activate na siya. So, pag mag-activate, it would be divided into two. Ang kaning 2, ang isa dire is kaning sa right side is for B memory cells. And ang purpose aning B memory cells is para the next time nga ma-expose siya sa isa ka specific pathogens, dili na siya dili na kay na siya maapektuhan and kanang rapid iyang pag-produce og antibodies. And next one here sa kaning left side is para mag-produce og antibodies as a primary response. Yes, so we're gonna talk about the primary response here. Wait lang, just colored. Wait lang, sure na good. Ayan. Here. Primary and secondary response. So, first one here, ang primary is initial response taking to 10 to 14 days for antibodies production. Why? Why is it Charis? Next one is secondary response. This one is the rapid or heightened response due to memory cells, also known as anamnestic or memory response. So, here's an explanation to that. The reason nga ang primary response is it takes to 10 to 14 days is because ang primary response occurs in our body pag ma-expose siya in the first time. It takes time before niya ma-recognize and before siya maka-produce og antibodies. For example, is when the person is infected with virus for the first time, is it takes 10 to 14 days. While ang secondary immune response is since na-recognize naman niya siya sa yung first exposure sa primary, sa pagka-secondary dayon is rapid na dayon kanang kuan na dayon niya kanang maka-response jud siya quickly. Okay, kasabutra no, kaya na tako na mo charing. Next one here, yes, the antibodies. We're going to talk about the antibodies. Antibodies are also known as immunoglobulins, are proteins produced by an antigen by lymphocytes in response to the presence of an antigen. All antibodies are in category of protein called immunoglobulin, as what I mentioned a while ago. Ang immunoglobulin is a globular glycoprotein in the blood that participate in the immune reaction found in lymph, tears, saliva, colostrum, mucous membrane, and blood plasma. Okay, so let's proceed to the structures of antibodies. The light and heavy chains, Fab and FC regions. So next one, ang basic immunoglobulin structures or the antibody structures resembles the letter Y. Two identical light polypeptide chains, two identical heavy polypeptide chains, and two antigen binding sites. And the last one is the fragments crystallizable region or the FC region. Grabe ang pag-pronounce na para bang okay, so here as we can see here na dire atong light chains between kaning color green dire and I right nga color green o kaning left nga color green mo na ang light chain, where ang heavy chain nato is nasa ubos gyud sa kaning Y. Alright. Okay. So, ang light chains is a fewer amino acid, shorter and lighter than heavy chains, while ang heavy chains is longer and heavier. So, in short, from the word itself, light, it means gaan jud siya, while bugat ang heavy chains. These chains are connected by disulfide bonds. Okay. Next one here is the antigen binding sites. The antigen binding sites are the monomer or bivalent or two binding sites. Bind specifically to antigenic determinants that stimulate its production. So, ang binding site nato is pangitaan natong binding site here, kani, and here. Mo na atong mga binding sites. Okay. Next one here is the role of FC region. The FC region enables the molecule to bind cells. For example, are the neutrophils, macrophage, basophils, and mast cells. Possess a surface receptor recognizing sites on the FC region. So, ang atong FC region is naa siya dire. As you can see here, at the FC fragments, dira na siya gapuyo, charis. Dira ang FC region na to. Okay, kasabot ra. So, next one here is antigen-antibody complexes. Mo ni siya, it is a form when our antibody combines with antigens. Their complexes are key to the immune defense system, and this leads to activation of leukocytes, or also known as white blood cells, lysis of bacterial cells, and enhanced phagocytosis. Next one is the immunoglobulin classes. So here we have the 5 main classes: the IgA, IgD, IgE, IgG, and IgM. So ang IgA, let's start with the IgA. The IgA molecular weight is 160 to 365 Dalton, and the total of 10 to 20% serum in our blood, and can exist as a monomers or a dimers. Ang a dimer is a two monomers held together by a G chain. Ang G chain is ang joint chain. Ang function ani is a predominant in saliva, tears, breast milk, and mucous secretions. Protects external openings and mucous membranes. Ang IgD is, di na ako basahan ng molecular weight. Ang function na lang ani is found makita ni siya sa surface of B cells may serve as an antigen receptors. IgE, the function is it involves the allergic response found in basophils and mast cells. IgG can cross the placenta, activates complements, promotes phagocytosis. While ang IgM functions is first antibody form in primary response, efficient at complement fixations. Alright. Next up is the antigen-antibody interaction and their effects. Okay, so this one is talks about how antibodies protect us. So first interaction is the prevention of attachment. So ang effect ani is ang pathogen coated with antibody is prevented from attaching to cell. So ang antibodies nato is ginabalot niya ang pathogen para dili siya makatouch sa atong cells. Next one is clumping of antigen. Antibodies link antigens together, forming a cluster that phagocyte can ingest. So, ang mga antibodies nato dire is dikit-dikit bitaw nila ang mga antigens para makaon sila sa atong mga phagocytic cells para ma-ingest sila. Next one is the neutralization of toxins. Antibodies bind to toxin molecules to prevent them from damaging cells. Next one is help with phagocytosis. Phagocytes can attach more easily to antigen that are coated with antibodies. So, syempre, since atong antibodies is gina-coat niya ang pathogen, so it can help for phagocytosis para ma-phagocytes, para ma-ingest atong mga pathogens dira. Then next one is the activations of complements. When complement attached to antibodies on cell surface, a series of reactions begin that activates complement to destroy cells. Alright. So, wait lang. Here, next, the last one is the activations of NK cells. NK cells respond to antibodies adhering to cell surface and attack cells. Alright. Actually, self-explanatory ra jud ni siya kaning antigen and antibodies interaction and their sites. What? Mo na to. The last one here, our monoclonal antibodies. Okay. Ang monoclonal antibodies is a purified antibodies directed against specific antigens and produced in laboratories using hydromas. We're gonna talk about hydromas here later, but first, we're gonna talk about the productions of monoclonal antibodies. Ang production niya is a single plasma cells that produce only one specific type of antibody is fused with rapidly dividing tumor cells. The resulting long-lived antibodies producing cells is called hydroma. So, ang hydroma naman po is ang hydromas are capable of producing large amount of specific antibodies. These antibodies are called monoclonal antibodies. Ang first monoclonal antibodies were produced in 1975, and the first diagnostic kit containing monoclonal antibodies was approved in the United States in 1981. So, here the uses of monoclonal antibodies. Okay. Commonly used in IDPs or immunologic procedure, are also known is to diagnose diseases, and it is being evaluated for possible use in fighting diseases, killing tumor cells, boosting the immune system and preventing organ rejections.
So let's proceed. So what is cell-mediated immunity? So this type of immunity is part of our body's defense system that mainly uses T cells or T lymphocytes to protect us from infections and abnormal cells like cancer. So definition and importance. First, CMI or cell-mediated immunity doesn't use antibodies. It uses T cells to defend the body. Second, It targets infected or abnormal cells inside the body. And last one, important for fighting viruses, cancers, and transplants. So cell-mediated immunity is different from antibody-based immunity. Instead of using antibodies, it depends on T cells to find and destroy harmful cells inside our body. And also, CMI is important in fighting viruses, cancer cells, and in rejecting foreign tissues like organ transplants. It helps keep our body protected from threats that hide inside our cells where antibodies can't reach. Three types of T cell. The first one is helper T cells or CD4. They activate other immune cells by releasing cytokines. So the helper T cells act like leaders. They send chemical signals called cytokines to activate other immune cells. The next one is cytotoxic T cells or CD8. They destroy infected or cancerous cells directly. The cytotoxic T cells are the killers. They directly attack and destroy infected or cancerous cells. And the third one is the regulatory T cells. They keep the immune system balanced and prevent autoimmunity. So the regulatory T cells are the peacekeepers. They make sure the immune response doesn't go overboard, preventing autoimmunity. So next slide, aside from T cells, we also have natural killer cells or NK cells. So the role of NK cells, first, they target virus-infected and tumor cells. Second, act fast, don't need prior activation. And the third one, they don't rely on MHC antigen presentation, unlike cytotoxic T cells. So natural killer cells are part of our innate immunity. That's why they act fast and don't need prior activation. And unlike cytotoxic T cells, they don't need to recognize an antigen through MHC molecules. They attack immediately once they detect something abnormal. How CMI works or how cell-mediated immunity works. So cell-mediated immunity works mainly in two ways. First, through cytokine secretion, and second, through direct killing. So through cytokine secretion, helper T cells release cytokines to alert and guide other immune cells. So here, helper T cells release these chemical messengers to alert and coordinate other immune cells. And second, through direct killing. Cytotoxic T cells release perforin and granzymes to destroy infected cells. So through direct killing, cytotoxic T cells recognize infected or abnormal cells and release substances like perforin and granzymes, which destroy the target cell. So for this part, we can learn something about humoral and cell-mediated immunity. So comparing the two immune defenses, so humoral and cell-mediated immunity are the two main types of adaptive immune responses. In humoral immunity, B lymphocytes produce antibodies that circulate in the blood and lymph to neutralize pathogens found outside the cells. This response is generally faster, especially after the body has already encountered the same antigen before. So because memory B cells quickly produce antibodies. On the other hand, cell-mediated immunity involves T lymphocytes, which directly attack and destroy infected or abnormal cells. This response tends to be slower at first, as T cells must recognize antigens presented by other cells before acting. But it provides a more targeted and long-lasting defense against intracellular infections and cancer cells.
Hypersensitivity reactions. This refers to an overly sensitive or overly reactive immune system. In such situations, the immune system, in an attempt to protect the person, causes irritation or damage to certain cells and tissues in the body. So what are hypersensitivity reactions? Some types of hypersensitivity reactions involve antibodies, whereas others do not. All types depend on the presence of antigen and T cells that are sensitized to that antigen. Hypersensitivity reactions is divided into two general categories. These are the immediate type and delayed type hypersensitivity reactions. Sa immediate type ito yung mga reactions na nag-occur from a few minutes to 24 hours after contact with a particular antigen. So under sa immediate type is type 1 hypersensitivity reactions, which is also called as anaphylactic reactions, or what we know as allergic reactions. Type 2 hypersensitivity reactions, these are the cytotoxic reactions, which involve the damage to or death of body cells. Type 3 hypersensitivity reactions, these are immune complexes reactions that damage to tissues and organs that is initiated by antigen-antibody complexes. The second category is the delayed type hypersensitivity reactions, which usually take 24 to 48 hours or longer to manifest themselves. The type 4 hypersensitivity reactions is under delayed type, which is also known as cell-mediated reactions, which are antibodies that play only a minor role, if any. An example is a positive TB skin test. So first, type 1 hypersensitivity reaction, which is called anaphylactic reactions. These include classic allergy responses such as hay fever symptoms, asthma, hives, gastrointestinal symptoms that result from food allergies, allergic responses to insects, insect stings, and drugs. And lastly, anaphylactic shock. These reactions all involve immunoglobulin antibodies and the release of chemical mediators, especially histamine from mast cells and basophils. So, the allergic response. People who are prone to allergies, tinatawag siyang atopic persons, produce IgE or also known as reagin antibodies when they are exposed to allergens. The immunoglobulin molecules bind to the surface of basophils and mast cells by their Fc regions. Chemical mediators of the allergic responses include histamine, prostaglandins, serotonin, bradykinin, which is a slow-reacting substance of anaphylaxis or also known as SRSA, leukotrienes, and chemicals that attract eosinophils, or also known as eosinophilotactic agents. So, ito yung process kung paano mag-occur yung allergic response. So, first, yung allergen is madidetect siya ng plasma cells. Then, yung plasma cells is it produces IgE antibodies which stick to either mast cells or basophils, and then when yung anti-allergen is mag-make contact siya sa IgE antibodies is magti-trigger siya ng release sa mast cells ng mga chemical mediators which include histamine, prostaglandins, serotonin, and bradykinin, and so on. So, next is localized anaphylaxis. So, yung type 1 hypersensitivity reaction is pwede siya maging localized or systemic. So, sa localized reactions is it usually involve mast cell degranulation, whereas systemic reactions usually involve basophil degranulation. So, the reason for this is sa mast cell kasi is usually makikita siya sa skin area natin. Whereas yung systemic reaction, which is the basophil degranulation, yung mga basophils kasi is mostly makikita siya sa bloodstream natin, which is, of course, nag-circle siya sa around our bodies. So examples of localized anaphylaxis are hay fever, asthma, and hives. So ang mechanism nito is when allergens, examples are pollen, dust, spores enter the body, they bind to the immunoglobulin on mast cells, causing the granulation and release of histamine, which leads to allergic symptoms. Antihistamines block histamine receptors to relieve symptoms like those in hay fever. Asthma involves other mediators beside histamine, so antihistamines are less effective. Food or drug allergens can cause reactions like hives, vomiting, and diarrhea. Next is systemic anaphylaxis. These are caused by basophil degranulation in the bloodstream. So it affects the whole body which is more serious than localized reactions. It may cause anaphylactic shock, which can be fatal. Common triggers for systemic anaphylaxis are drugs, examples are penicillin, or insect venom. Mechanism of this systemic anaphylaxis is yung allergen, magbabind siya sa immunoglobulin E on our basophils, and then magkakause siya ng degranulation, and of course mag-release siya ng massive histamine release. Symptoms of this sa early age is itching, flushing, swelling, difficulty breathing, and later low blood pressure, nausea, vomiting, cramps, and unconsciousness. Treatment for systemic anaphylaxis are immediate epinephrine or adrenaline and antihistamines. Prevention is always check for drug or insect allergies. Allergic individuals should wear medical jewelry. So, latex allergy, ito siya is example siya ng type 1 hypersensitivity reaction. Ito is mostly common siya sa atin na mga healthcare practitioners because of the frequent exposure to latex gloves, yung pag-wear ng gloves and other medical products. Studies show that about 1 to 6% of the general population and 8 to 12% of healthcare workers are sensitized to latex. So latex can trigger 3 types of allergic reactions. The first is the irritant contact dermatitis, which is most common reaction. It causes dry, itchy, and irritated skin, usually sa ating hands. It is not a true allergy because immune system is not involved. So hindi siya really type 1 hypersensitivity reaction. So number 2 is allergic contact dermatitis, which is caused by chemicals added during latex processing. Ito siya is a delayed hypersensitivity, more on type 4 reaction. Symptoms resemble poison ivy, redness, itching, and rash. So number 3 is the latex allergy. Ito siya is ang true type 1 immunoglobulin mediated hypersensitivity. It is caused by sensitization to latex proteins. And ito naman yung mild symptoms, redness, hives, itching, and then the severe symptoms, respiratory distress, asthma, or anaphylactic shock. It is diagnosed by antibody test or skin testing, and the best prevention is to avoid latex exposure and use latex-safe environments in healthcare settings. So next is the type 3 hypersensitivity reactions. So type 2 hypersensitivity reactions are cytotoxic. So meaning the body's own cells are destroyed. Yung cells natin mismo ang nasisira. So they involve immunoglobulin G or immunoglobulin M and the complement system. Examples of these are incompatible blood transfusions, Rh incompatibility, and myasthenia gravis. So a typical type 2 hypersensitivity reaction might follow this sequence. Ito yung sequence niya. Step 1, a particular drug binds to the surface of a body cell. The anti-drug antibodies then bind to the drug, and then this initiates the complement activation on the cell surface, and then the complement cascade leads to lysis of the body cell, so destruction of the body cell. Next is the type 3 hypersensitivity reactions, which is also known as immune complex reactions kasi mostly nag-involve siya ng ating immune complexes. It is caused by antigen antibody or immunoglobulin G or immunoglobulin M. These complexes that deposit in tissues, these are complexes that deposit in our tissues. It activates complement and attracts neutrophils, leading to inflammation and tissue damage. Examples of type 3 hypersensitivity reactions are serum sickness, which is an antibodies to horse serum proteins from immune complexes. Symptoms include hives, fever, kidney, and joint tissues. It occurs in 10% of patients given horse anti-serum. Another is autoimmune diseases, which are systemic lupus erythematosus or SLE and rheumatoid arthritis. Another, lastly, is the post-streptococcal complications, which are, ito yung pinaka biggest example, which is ang immune complexes is mag-deposit siya sa ating heart, which causes rheumatic heart disease, and then joints, which is arthritis, and then kidneys for glomerulonephritis. Next is the type 4 hypersensitivity reactions, which is also called delayed type hypersensitivity reaction because nasabi natin kanina is under siya sa DTH reaction, or also known as cell-mediated immunity reactions. So, since delayed type siya, it occurs 24 to 48 hours or longer after exposure. It is seen in tuberculin and fungal skin tests, contact dermatitis, and transplant rejection. Defense siya against intracellular bacteria and fungi. It involves macrophages, cytotoxic T cells, and NK cells. So, unlike sa other, yung type 1, type 2, type 3, here antibodies are not involved. Example of type 4 hypersensitivity reactions is the positive TB skin test, or also known as Mantoux test. This is a test that uses tuberculin or PPD from Mycobacterium tuberculosis. It is injected intradermally. If a person has immunologic memory, a DTH reaction occurs, redness and swelling, wheal and flare. So, here are the steps of paano nag-occur ang type 4. 2 to 3 hours, influx of polymorphonuclear cells or PMNs. Next is the PMNs disperse, lymphocytes and macrophages arrive. And then 12 to 18 hours, area becomes red or erythematose and then swollen, also known as edematose. And then to 24 to 48 hours, maximum intensity. Dito na intensify yung redness and swelling. And then afterward, magsiswelling. Yung swelling and redness is magpe-fade siya as lymphocytes and macrophages disperse. Here is the interpretation of a positive TB skin test. It may indicate any of the following. First is the active TB disease, chest x-ray positive, coughing, sputum with acid-fast bacilli. Number 2 is the past TB, when you are recovered na person previously had TB. And then number 3, past exposure, cleared by immunity, no live bacteria, still given 6-month isoniazid. Number 4, latent TB infection, which harbors bacteria without symptoms, which is also treated with isoniazid. Number 5, lastly, BCG vaccination history, which is received Bacillus calmette-guerin vaccine, may cause false positive. So what is a BCG vaccine? It is made from attenuated Mycobacterium bovis, which is 50% effective in TB prevention, tuberculosis, which also causes temporary positive skin test. Other type 4 reactions are contact dermatitis like metals, poison IV, cosmetics, topical drugs, and also transplant rejection due to cytokines and antibodies reacting with foreign tissue and antigens. Autoimmune diseases. These are diseases that occur when the immune system attacks the body's own tissues, mistaking them as foreign or non-self. It may result from tissues not exposed to the immune system during fetal development. Examples are the lens, brain, spinal cord, and sperm. Later exposure, injury or surgery to antibody formation, to tissue destruction. Examples are blindness, encephalitis, and sterility. Certain drugs or viruses may alter host cell antigens, triggering autoantibody or sensitized T-cell reactions. Over 80 autoimmune diseases identified and affects 10 million people in the US. So here are the classification of autoimmune diseases. First is organ-specific. It affects one organ or tissue. Examples are thyroid, Hashimoto thyroiditis, Graves' disease, primary myxedema, thyrotoxicosis, and in the stomach, pernicious anemia, in the adrenal glands, Addison disease, and in the pancreas, type 1 diabetes mellitus, which is insulin dependent. Number 2 is non-organ specific. It affects multiple systems like like in myasthenia gravis, our muscles, dermatomyositis, our skin, systemic lupus erythematosus, SLE, our kidneys, lungs, skin, brain, scleroderma, skin, lungs, kidneys, gastrointestinal tract, rheumatoid arthritis, which is our joints. So here are the hypersensitivity types involved. Our type 2, which is myasthenia gravis, and then in our type 3, rheumatoid arthritis, and then SLE, and then for our type 4, some cell-mediated autoimmune responses. Next is immunosuppression. Immunosuppression is a properly functioning immune system, which can be called immunocompetent, and then a malfunctioning or weakened immune system is called immunosuppressed or immunodepressed or immunocompromised. Causes of immunodeficiency, most common cause worldwide is malnutrition, and then second is acquired immunodeficiencies. These are caused by drugs like chemotherapy, transplant medications, and irradiation like radiation therapy, and then infectious disease, especially HIV infection. So in HIV infection, HIV destroys T cells, leading to decreased antibody production, causes inability to fight off pathogens. It results in AIDS. AIDS patients often die from opportunistic infections like viral, bacterial, fungal, and parasitic. And then the third causes of immunodeficiency is aging. The decline in T cell regulation reduces antibody production and increases infection risk in the elderly. So next one is the inherited or genetic immunodeficiencies. These are caused by defects in antibody production, complement phagocytes or NK cells. Examples of these are the chronic granulomatous disease or CGD, which is a defective phagocytic killing. Next is Celiac-Higashi syndrome, an abnormal lysosomal function. Number 3 is the severe combined immunodeficiency or SCID, which is the absence or dysfunction of B and T cells. It leads to severe recurrent infections. And then number 4, DiGeorge syndrome. It is the congenital absence of thymus and parathyroid, causing frequent infections and delayed growth. Fifth is the Wiskott-Aldrich syndrome, which is the deficiency of B cells, T cells, monocytes, and platelets, which causes bleeding, eczema, recurrent infections. Treatment and advances are bone marrow transplantation and gene therapy, which show promise in treating genetic immunodeficiencies, and then ongoing research from the Human Genome Project, which aims to improve understanding and treatment options. Lastly is the antibody deficiency disorders, agammaglobulinemia, which is the absence of gamma globulins or antibodies in blood. It leads to extreme susceptibility to infections. Treatment are bone marrow transplant to restore immune function. And then last, hypogammaglobulinemia, which is low antibody levels, less severe than agammaglobulinemia, which leads to delayed recovery from infections. Bruton's hypogammaglobulinemia are hereditary with few or no circulating B cells.
Immunology laboratory and clinical applications. The immunologic procedures include tests to diagnose infectious disease and immune system disorders. It also determines tissue compatibility for organ and tissue transplants and detect and measure various serum components. Historically, the amount of time it takes to get the laboratory result has been the most criticism of the CML or the clinical microbiology laboratory. Sometimes days or even weeks are necessary to isolate pathogens from clinical specimens to get them growing in pure culture in large numbers, and also to perform the tests necessary to identify them. Immunodiagnostic procedure are laboratory procedure that help to diagnose infectious diseases by detecting either antigens or antibodies in clinical specimens. The result of such procedures are often available on the same day that the clinical specimen is collected from the patient. IDPs performed on serum specimens are called serologic procedures. Some IDPs are designed to detect antigens, whereas others detect antibodies. The detection of antigens in a clinical specimen is an indication that a particular pathogen is present in the patient, thus providing direct evidence that the patient is infected with that pathogen. There are four possible explanations for the presence of antibodies to a particular pathogen. First, the present infection. Or the example, the person is currently infected with the pathogen. Second, the past infection. This is where the person was infected with the pathogen in the past and antibodies are still present in his or her body. Third, the vaccination. The antibodies are the result of the person having been vaccinated against that particular pathogen at some time in the past. For example, a person's serum may contain antibodies against influenza viruses because he or she received a flu shot last year. And lastly, the cross-reactivity or the cross-reaction. Although we consider antibodies to be very specific, there are instances where the antibodies can react with epitopes that are molecularly similar. The agglutination procedure. There are two types, the schematic representation and the actual lattice agglutination test. The schematic representation illustrating how antigen can agglutinate antibody-bound lattice beads. This results in clumping of the lattice beads, while the actual lattice agglutination test illustrating positive results or the clumping of lattice beads and the negative results, the no clumping of lattice beads. Antigen detection procedures is the clinical specimen mixed with a particular antiserum. For example, a drop of cerebrospinal fluid from a patient with meningitis is mixed with a drop of antiserum containing antibodies against the Haemophilus influenzae. Agglutination procedure used for ABO blood typing. The clumping of the person's RBCs with either anti-A antiserum or anti-B antiserum signifies that A antigen or B antigen is present on his or her RBCs. For example, a drop of serum from a patient suspected of having Lyme disease is mixed with a suspension of Borrelia burgdorferi, where the Borrelia burgdorferi is the bacterium that causes Lyme disease. A visible antigen-antibody reaction is evidence that the patient's serum contained antibodies against Borrelia burgdorferi and the patient's condition is diagnosed as Lyme diseases.